me about it. Tell me about it. It's on Tuesday, Thursday, all week. All those activities that I encourage my daughter to do, but now I regret because I didn't understand it meant that I would become a driver.
All right. All right. Well, once I cease being mesmerized by this video of the flock of birds, it will be my pleasure to introduce André. So André Fenton, I guess he got asked the question, how do we know each other? And we do indeed go way back. So André is trained as a neurobiologist. Went to McGill and then did a stint with Jan Buresh. And Buresh was a pioneer in the use of methods for reversible localized inactivation based on this principle of depression, spreading depression following epilepsy. And so André came in kind of trained in the methods of behavioral psychology, but then did his graduate work with Bob Muller at SUNY Downstate. And Bob Muller, I was working with Bruce at the time as a postdoc. André was doing his graduate work with Bob Muller, and Bob Muller being the most authentic New Yorker that I think one could actually imagine. He's sort of a caricature of the sort of the grizzled but authentic New Yorker. And André did his graduate work looking at at place field control, Q control of place fields, kind of establishing the basis for linking cognitive maps to behavior. So using old school behavioral electrophysiology, reversible lesions, physiological mapping. He then went back, following his graduate work, went back to Prague, where this was the Academy of Sciences, Czech Republic in Prague, where Buresh had been working. He had his research lab there for a number of years before he came back to the States, back to SUNY Downstate, where he established a lab. And it was at that time where he really kind of expanded doing the work, hippocampal place fields, electrophysiology, behavior, but also linking to synaptic physiology. And there was a paper, this was in 2006, working with Todd Sachter at SUNY Downstate, and Eva Pastelkova, she was a postdoc with Yuri at the time, was she? I think so, yeah.
First me and then Yuri.
Yeah, you and then Yuri, right. So, and this was identifying a protein kinase C isoform, this was PKM zeta, that was linked to long-term plasticity. So identifying kind of molecular mechanisms involved in persistence of synaptic plasticity and memory, combined with behavior and electrophysiology. And so that's what he worked on at Downstate until in 2010, I guess it was, you moved to NYU, Center for Neuroscience, and kind of established a very successful lab. They're so successful that they turned to him as the director. And so since last year, I guess, he's been the director of the Center for Neuroscience there and also the chair of the department, and overseeing the vast kind of sprawling expanse of scientists there and graduate students involved in the graduate program. So it's a real pleasure to have André here. Our trajectories have sort of followed from the old timers back at the time. I was working with Bruce McNaughton, André with now the late Bob Muller, and Jan passed as well. Buresh passed about 10 years ago. So
That's who have trained me.
Yes. So the new generation, kind of thinking about how mechanisms of plasticity, the electrophysiology of the hippocampus, and the organization of synaptic ensembles manifest in coordinated behavior. and high-level cognitive function as it might relate to neuropsychiatric and neurocognitive function. And so it's always exciting to hear what Andre's doing. Andre?
Thank you, sir.
Good to see you.
See if I do this right. Can you all hear me? Okay, great. So thanks for the introduction, Matt. It's been a fantastic day of conversations. I'd like to have the conversations continue, so feel absolutely free to interrupt and ask for clarifications or even challenge anything up here. No reason to be polite, because I was trained by Bob Mueller. He was not polite. So I'm going to talk today about a number of things in a coordinated way, hopefully, about some of what we've learned about memory, in fact, that neural circuits can learn to learn, and mostly about how neural activity gets coordinated, and in the case of the Fragile X mutant mouse model, discoordinated in controlling cognitive representations. And you'll see the talk has more or less 4 parts. These are my disclosures. None of them are relevant for the talk. So this talented group of graduate students and postdocs will start us off. And they really were trying to understand, this was their first time looking at the Fragile X model mouse, the FMR1 null mouse, and they were trying to understand, what goes wrong with those neurons in the freely behaving mouse, and I'll tell you some of what they learned. So by way of small introduction, I don't think I need to delve into this. Fragile X syndrome is the most prevalent or most common single gene cause of intellectual disability and autism spectrum disorder. About 30% of these individuals have features of autism. 15% of them have seizures. The impairments are mostly seen in males because it happens to be X-linked, and obviously because of that, it's more prevalent in males. What is the disorder? What is the genetic defect? There's a trinucleotide CGG repeat that we all have in the FMR1 gene, and if that repeat expands over time and you happen to have more than 200 or so of them, then the gene gets silenced. And because the gene is silenced, you don't have any messenger RNA, and so you don't have the FMRP protein. What does that protein do? Well, it turns out we know very well what the protein does. It's a repressor of translation. Generally, maybe about 400 or so transcripts are repressed from being translated when this is around. But nonetheless, what you'll see here is if we look in our hippocampus at Ph.D. 95, you can see here in the control animals, there's less Ph.D. 95 than in the null animals, which is what you would expect from a translation repressor. But here's my favorite molecule, PKM zeta, and you can see that that's actually decreased, interestingly, in the FMR1 null mouse. So it's not at all straightforward whether you get more or less of this, despite the protein being a very clear translation repressor. Here's a nice paper that was done in 2017, where they actually looked at the translating transcripts, and you can see that some are upregulated and some are downregulated. So it's not at all clear, despite this very particular and selective manipulation. One gene is being silenced, but the consequences are difficult to predict, and they're multifold. And so that presents a scientific challenge, right? On the one hand, we know it's absolutely clear that FMRP, the protein, is repressing protein synthesis. And on the other hand, we know, very specifically that it causes intellectual disability and autistic features. And it's our job, in a sense, to go from these two really quite clear facts and somehow cross that gap. And I'm going to try to do that, or at least give you some insight into our attempts to do that. So how do we do that? It turns out to be difficult. Why? Because there's no clear and simple causal chain to go from the genetics, if you will, to the social deficit, social behavior. And I like to use this cartoon of the various levels of biology. Obviously, they're not all here. And you can see that these arrows, these, if you will, causal or interacting arrows, go in both directions. It's not clear how behavior is affecting the brain, or the brain is affecting behavior. The individual behavior affects what the social group does, and so forth. And so this presents a very difficult challenge. How do we meet that challenge? Well, I'm going to describe today our attempts to do so, not by, importantly, not by trying to mimic this disorder in any way. I will not claim, or I don't want you to think, that these mice have Fragile X syndrome. They don't. They have a mimic of the genetic defect, because we'd like to understand what the elimination, if you will, of the protein, you can imagine that being here, how that actually causes consequences. And there are many ideas about this, and we'll test some of those ideas. And so while these animal models might be, in some sense, a lousy mimic of the human condition, they're quite powerful when you think of them not really as subjects that have the disorder, but as reagents, if you will, that we're going to use to test ideas about the disorder. And that's the way I want to approach this. So one of the very clear general hypotheses is that, there's, and I'll show you some of the evidence for it, that some kinds of synaptic dysplasticity, dysregulated changes in synapses, disrupts the neural information processing when the FMRP protein is lost. And so that would predict, as an example, that the place cell tuning of these individual neurons in FMR1 null cells would be impaired in some way. They'd be unusual in one way or another. And you can find this sort of idea in many places. And we are going to evaluate that at the beginning. I also want to point out another hypothesis, which is one I've developed over the years, what I call the discoordination hypothesis. And when you actually look at the mechanisms of plasticity that have been altered, you can see that the individual cells might actually have ways of maintaining their individuality and their individual tuning properties, but the network as a collective might lose its ability to coordinate in appropriate ways. I'll unpack these later, but these are the two hypotheses, if you will, that we're going to try and evaluate using this animal model. A little bit of introduction to the mice. They happen to be susceptible to seizures. When they're young, you play a loud noise like an alarm clock, then you'll see that they will run wildly, and in fact, the majority, almost all of them, will have a seizure, so it's an audiogenic seizure. They have social deficits, if you will. So they will prefer, just like a regular wild-type mouse, to investigate another mouse over an inanimate object. If you change the mouse, then they will not prefer the novel mouse, whereas a wild-type mouse would, and that's only after a 5-minute delay. But if you wait 24 hours and bring a new mouse, then they're very much like wild-type mice, in that they will investigate preferentially the novel mouse. This is a task I'm going to describe in some detail because we'll use it a lot. This I call the active place avoidance task. And what you can see here is a mouse on a rotating arena. The arena is rotating in this video 5 times faster than real. Otherwise, it'd be really boring to watch. And the mouse's job is not to get detected in the area that's marked red. The area that's marked red is marked red for you and me so that we can see it. It's not marked for the mouse. The mouse can look around the room, however, and figure out where that area is because there are cues hanging on the walls and so on. And that area is going to be stationary relative to those cues. And you can see this mouse is doing a pretty decent job of staying out of there. Why does it stay out of there? Because if it's detected in there, the computer turns on a half-second mild foot shock. It's only 0.2 milliamp. So the animal doesn't freeze. It doesn't ever show some kind of fear response. In fact, its cortisol levels, or corticosterone levels rather, don't even elevate compared to walking around the maze in these tests. And so what we can measure is how well the animal avoids that unpleasant place. And if you do that, you can see that in pre-training, when there's no shock, the animals will enter that shock location, say, 15 or 20 times in 10 minutes. Once you activate the shock, then they tend to not enter there, and the learning shows that they tend to enter maybe one or so times over time. And they'll remember this for at least a month, in this case, just one day. And what's, I hope, very clear here is that these animals, the FMR1 null animals, are no different. They're certainly not worse than the wild-type animals in being able to learn this. We can test whether or not they actually have what I'll call cognitive flexibility, because we can change the location of the shock zone and now ask them to avoid the place that they had preferred, because that's now being shocked. And so the animals actually have a very robust and reliable cognitive inflexibility. You can see here that the wild-type animals very rapidly learn to avoid that location. And these animals with the missing the FMRP, they do so more slowly. In fact, I can show you another variant of this. If we instead, after this conflict learning, or instead of the conflict learning, we simply turn off the shock. And so now the animals extinguish this is the amount of time they spent in the shock zone. They're no different from controls here on the first session, but if you wait 2 hours and bring them back, you'll see here, while the wild-type animals actually now are willing to spend time in the shock zone, these guys are not avoiding at all. They're relearning, if you will. So they have this inflexibility, and the way I describe it is they have an impairment in learning something that contradicts what they had initially learned. And that'll be an important idea to come back to. You might think, well, it's not quite so sophisticated, Andre. I want to just show you a little data. What I can also measure is the number of times or the amount of time it takes the animals to enter for the first time, second time, third time, and so on, up to the 5 times it enters the shock zone as learning. And you can see here that the wild type and the knockout animals are learning this approximately the same. The key in this experiment here is that you'll see that after this initial training here, just like I showed you before, I'm going to add another shock zone, okay? But it's not going to be in conflict with what the animal had learned. So I'm adding here a blue shock zone that is a rotating shock zone. So as the arena was rotating, a part of the floor that's rotating with the animal will rotate with the arena, and the animal will also have to avoid that location. You can see there's no way to distinguish the two groups here. What about in avoiding the rotating arena? Again, no impairment. So their deficit is really quite particular, quite specific to things, I would argue, that contradict what they had initially learned. As far as I and the literature on these mice can show, they're really pretty good at learning almost anything. What they're bad at doing is learning something that contradicts what they had initially learned. So what else do we know about these mice? In Mark Baer's lab, you can see here what the main deficit is. What I want to point out first is, as others have shown, if you try and induce LTP in slices from these animals in the Schaefer collateral pathway, for example, there's no difference in the LTP expression or the induction, early or late LTP in these mice. However, if you try and induce LTD using group 1 mGlu-R stimulation, an agonist called DHPG, for example, you will get a depression, and the depression is long-lasting, and you can see the magnitude of that depression is greater in these knockout mice, as Mark Baird showed. And some to-do has been made about this because the mGlu-R theory of FXS has developed from this, which asserts more or less that the mGlu-R stimulated translation pathways are dysregulated in these animals. As I showed you, the translation was abnormal, and that's actually the cause, if you will, of all of their various phenotypic problems. So we'll address that. I just put this here to remind you that the synaptic plasticity looks, the expression of Ph.D. 95 actually does what you'd expect from a loss of repression. It goes up, and however, PKM zeta expression, another synaptic plasticity molecule, happens to go down. If we do the same experiment as I showed you before in these animals, taking them from the home cage, we again get the exact same result, that the animals are quite normal in their expression of synaptic strength as a function of stimulation. If we try and induce LTP, just as I showed you before, in our hands it's the same. There's something interesting to think about here, though. These animals, as is done standardly, are just taken from the home cage. They haven't learned anything. We haven't challenged them in any way. We've just, plucked them by the hand of God from the cage to be offered up as hippocampal slices. Okay. What happens, however, if these animals are allowed to be trained in the place avoidance task that I showed you, or exposed, that is put in the same condition, but the shock is never turned on, compared to the home cage or naive? You can see here in wild-type animals, if they've been exposed, they've just walked around, they're no different than naive. After the training, then that pathway, that synaptic pathway strengthens, right, for a particular stimulus. You have a stronger synaptic population response. But look at the knockout animals. Just walking around on the arena for I think this was 30 minutes, their synaptic pathway has enhanced a lot more than the wild-type pathway, and it's not actually very different from what happens if they actually get trained in this task. So they have, if you will, a hyperplasticity, or at least a not normally regulated plasticity. That is experience dependent, okay? And that will be important. What about the LTP? Well, an interesting phenomena that I'd love to talk about, but I won't dig into. Let's look at the wild-type animals. After this training in the place avoidance task, if you induce LTP in these ex vivo slices, you get LTP, but the magnitude is substantially less, significantly less than if the animals were naive or simply walked around on the arena. I won't dig into this, but if you're super interested, the reason for this is not that the synapses have saturated in any way. It becomes actually easier to depress this pathway and harder to potentiate it, and we can check that by actually repeating the high-frequency stimulation, and it will go up to the wild-type levels eventually. Look at the knockout animals, however, the same pattern exists. You'll notice that the magnitude of the LTP is about twice as much. And the naive animals and the exposed animals, again, have this very high exuberant LTP, and there's still an attenuation if the animals have the training. So the pattern is the same, but there's a lot more if you will, synaptic plasticity that we can induce and that we can observe after the animal has had its own experience. So that's sort of a characterization of the animals. So Ein Chung, Dino Dvorak, and Elliot Levy in the lab were very interested in what the animals are actually learning to do with this task. Are they really just learning to avoid a place, or is there something more interesting going on? And if you think about it, while the animal's on this rotating arena, it has to do something more than just recognize where it is and avoid the shock zone. It also has to ignore where it happens to be on the irrelevant, if you will, rotating floor, because it's being shocked at locations on the floor that change with respect to the actual place that the shock is organized in, which is in the room. And so I'll show you very briefly a subset of the results from this endeavor. Before I show you those results, I want to show you that we actually, by studying this particular set of cells, are studying an important group of cells and an important set of synapses for this task. Here's a trap mouse where we're using the immediate early gene ARC, and what we're going to do is tag the neurons that were active, or at least stimulated ARC expression, when the animal was remembering this task. And so they get to express EYFP as well as channel rhodopsin. And you can already see that the expression levels are not homogeneous in the hippocampus. That's actually an interesting line of study, but I won't focus on that. What we're able to do, though, and that's what Claudia Joe is able to do, is that she can optogenetically activate these cells, in this case a month later, when the animal is in a novel environment. When the animal's in a novel environment, you don't turn the light on, the animals just walk around everywhere. What happens when she put the animal in this video for the very first time in its life, it's in this room, it's never been in this room before, never been on a rotating arena that looks this way before. She's, I think it's at 4 or 10 hertz, I can't recall, we do both. Pulsing the light, and you'll see that the animal actually avoids this particular region of the space. It has no explicit reason to do that other than presumably the light is activating those cells for it to avoid. And so in some sense this shows that these cells, or at least the reactivation of these cells, is in a sense sufficient for this, for the expression of this avoidance. And here's a summary of where this animal went in this environment in which it's naive. There's a whole line of research that we do continuing from this, but I just wanted to show it to you to show that these cells are actually really important, in some sense sufficient for this avoidance, because we're going to study those cells. What else can I say? And here we'll use miniscopes and do some calcium imaging while the animals are doing this task. And if we do that, what we can see is, yes, these cells express firing fields like place cells would be expected to. More importantly, however, if you calculate what I call positional information, which you can think about as how much information does the current spike train, and that current spike train in this case is, I think, 300 milliseconds across the ensemble, how much information does it tell you about where the animal is in the room compared to how much information it tells you about where the animal is on the rotating surface? And if you take the difference of that across the ensemble, you'll see that the activity tends to fluctuate back and forth for a few tens of seconds, signaling the animal's location in the room in red, or the animal's location preferentially in blue. And this in black here is just the total firing of the population. So the activity is fluctuating back and forth exactly as you might imagine if the animal was using something like cognitive control, which you could operationally define as judiciously representing two classes of information according to the animal's particular needs. What about the animal's particular needs? If you look here, you can look here. What is plotted here is the likelihood that given where the animal happens to be, that you'll be in the room frame. Yellow indicates high likelihood. So on day one, before there's any shock, that there's no organization to that, and you can see that when the animal's close to the particular shock zone, there's a yellow. When it's far away, it's blue. So when the animal's far from the shock zone, it happens to represent this space in the arena frame, and when it's close to the shock zone, it's representing this space in the room frame. And so this fluctuation is purposeful, and in some sense, or in an explicit sense rather, this task is causing the hippocampus to do this judicious switching, and I will come back to that in a moment. So what does this task actually cause the animals to do? And Chung decided to find out. And so we created three variants of the task. Okay? The three variants of the task took place in the exact actual environments. They all looked like this. The three variants, however, can be thought about in this way. This is the standard task. We call it the cognitive controlling condition, cognitive control training condition. This is what you've been seeing. The place learning condition is exactly the same task, except we put a thin layer of water on the arena surface. And we put the water there because we know that it's the olfactory cues on the arena surface that define where the animal is on the arena surface. And so by dissolving them and making them unstable, the animal in some sense doesn't have to ignore where it is on the arena surface. And this spatial exploration control, it's what I've shown you before, is the animal in the arena. The shock is just never on. These pictures are misleading because they show you that it looks different. If you think about it, the shock is, the animal only experiences the shock if it's in the shock zone, and when it does so, it's less than 0.5% of the time in the arena. So these are very, very similar physical experiences. What we can observe if you train animals in one environment, as I just described in the initial condition, and you wait a week, okay, and test their retention in that environment, they will have learned it. I won't bother to show you that. The interesting thing is, if you wait another few days and put them in a novel room, like I showed you with the optogenetic activation, and train them and see what will they learn in the subsequent training, what's interesting is that they actually learn better, although there's nothing physically the same in these two environments. They learn better with the subsequent training as just shown here, right? This is the time it takes to enter the first, second, and so on to the fifth time. What might be more compelling is if you look down here, we can do the same thing and train the animals in the initial conditions in the spatial exploration or in the place learning situation, and as well as this CCT trained group. And you can see that the CCT trained group actually learns in the novel environment better than these other control groups, despite actually having done pretty much the same thing, including the animals that had the place training. And so this is actually because the animals have been forced, if you will, during training to ignore those irrelevant cues on the arena surface. And lastly, just to convince you that the animals have actually learned not only this place avoidance, we can put them in a T-maze after the initial training. We wait 1 or 2 weeks, and we put them in a T-maze in a different part of the room, and we ask them first to go right, and then after 10 trials to go left, and then another 10 trials, then to go right. And you can see in the initial block of 10 trials, there's no difference between the groups, but in the second block of 10 trials, when the animals have to do the opposite of what they had initially learned, the animals that have the cognitive control training are improved compared to the other groups. So something happens during this task that changes the way the animals are going to behave going forward. What happens? Well, we did the following experiment. We repeated that training, this time with electrodes in the across the hippocampal circuit, and I'll describe what happened in the medial perforant path input to the dentate gyrus in the upper blade of the dentate gyrus. And you can see here, if we stimulate and measure the synaptic response at the dentate gyrus in the upper blade, that this input weakens quite substantially. Notice it's not potentiated, but weakened after that cognitive control training. This is the quantification from the curve. But it doesn't change if the animals have only had the spatial exploration or the place learning version. So again, this training causes some kind of long-lasting change. This kind of change actually lasts at least 60 days. I don't think I'll show that to you, but if you look in the paper, we can come back and see that these changes maintain for a very long time. If you do the same experiment with our FMR1 null mice, this is what I just showed you, that the stimulation after training in the wild-type mice, the pathway is depressed in the upper blade of the dentate gyrus, but not in the lower blade of the dentate gyrus. And we're quite sure about localizing this because we're using current source density to measure these changes. But you don't see that change in the knockout mice. Right? There isn't this plasticity, at least in the depression in the upper blade, but it's quite the opposite. You get the expression of the plasticity change, the inhibition, actually in the lower blade of the dentate gyrus. And I'll be happy to describe what the different innervation schemes are for the upper and lower blade. But suffice it to say, again, there are changes. They're just not the normal changes, or at least the wild-type changes in these animals. And recall, these animals are impaired in their ability to do this task, not when they initially learn it, but when they have to learn a version of it that is contradictory to what they had initially learned. So just in summary, what have I shown you in that series of experiments? That this kind of training, this cognitive control training, if you will, right, causes a kind of learning to learn in the wild-type animals, not so in these FMR1 animals. Remember, they're impaired in the conflict version. It causes a persistent decrease in this input from the medial perforant path. I can just tell you a similar thing is happening in the direct input from the entorhinal cortex to CA1. I just haven't described that to you. And the way we think about that computationally is that, there are inputs to this network, and here's a little model simulation that would show that happen to be diverse. This kind of change is going to remove some of the noise. And I won't give you the evidence for it, but feel free to look in the paper, because the cool part of the paper is that we identified a disinhibitory circuit such that these surviving inputs are actually enhanced. And so the signal to noise in this system is persistently enhanced, and that actually explains the learning to learn in the wild-type animals. But these changes are not observed in the FMR1 null mouse in the same parts of the circuit. You get a different set of changes, those in the lower blade of the dentate gyrus rather than in the upper blade of the dentate gyrus. So it's like these mice are doing kind of the right things, but they're not doing them in the normal places, at least in this circuit. And that is a theme I will continue with in the next piece. So what have I shown you with these mice? They're characterized by, a dysregulated protein synthesis. They have pretty normal baseline synaptic function, anywhere we have measured as well as others. They have a robust experience-dependent abnormality in how synapses change, as we and others, as we have measured. And they have, frankly, not very strong hippocampus-dependent behavioral deficits. They're there. You have to look for them, and they're very particular. So I want to talk a little bit about place cells. And before we talk about place cells, because if you remember, the main hypothesis we wanted to test makes a prediction about how those cells would behave given this set of dysplasticity, the fact that the dysplasticity is experience dependent, and so on. Before we jump into the data, I want to describe, I probably don't have to do that here, but this is a place cell I recorded when I was a graduate student. So a very old video. You can hear the amplified action potentials of likely one cell. I've drawn where the cell fires when you hear that. And so mostly the activity is when the animal's over here. There's a little bit of activity here. So we can record these cells, and the goal is to record many of these cells. You know, in 2014, John O'Keefe shared the Nobel Prize for his discovery of those cells with the Mosers, who found a similar cell type, these grid cells. And the reason I mention this is because this prize, in many ways, I think is the way it's talked about is misleading, at least for the purposes of this talk, and maybe in general. It was described as the brain's GPS. Okay, so that's great, it's 2014. But if you go back to when John O'Keefe first found these cells and wrote about these cells, and you'll see that he wrote about them, I think, very eloquently and compellingly in this book with Lynn Nadell. And let me just read for you the second sentence in the book. Okay? We shall argue that the hippocampus is the core of a neural memory system, providing, and here's the important part, an objective spatial framework within which the items and events of an organism's experience are located and interrelated. And later on, 20 pages later, it said, they write, space was a way of perceiving, not a thing to be perceived. So from their point of view, okay, and this is a point of view that endures, but the field doesn't typically think about, these cells are creating a infrastructure, if you will, within which the animal can understand its experience, rather than simply reflecting the stimuli in the world where the animal is. And from that perspective, I'd like us to think about how these cells operate in the FMR1 null mice. I was going to philosophize here, so I'll skip Kant. Not necessary, but since I have a background in philosophy, I would love to do it. Right, so I want to remind you that we're using these animals to test a prediction of what you would expect from this place cell system, given the plasticity alterations that I had described. Right, so the disruption hypothesis would predict that given these plasticity features, that the neural information processing is going to be abnormal in these animals. And in particular, since the plasticity disruptions happened from birth, right, all through development and into the animal's adulthood. And, very much, we can ignore the analogy here, for time, but these, what you'd expect is abnormal place cell firing of these individual cells, in particular because all of the plasticity that's supposed to be part of the memory system, that's supposed to be part of the organization, isn't going to be well coordinated. Well, the surprising thing to us from one point of view, at least from this hypothesis point of view, is that the place cells are absolutely normal in these animals. Here are a bunch of wild-type place cells when the animals were recorded in a box, and here are the similar cells recorded while the animal from the FMR1 null mice. You can't tell them apart. If there is a difference, if you dig into the paper, it's actually these place cells look a little bit better than the wild-type place cells. And they have plenty of interneurons, and they seem to make the proper spatial tuning patterns and so on. If you look at the individual properties of these cells, do they fire in bursts? Do they not fire in bursts if they're interneurons? What are the biophysical properties of the cells? What is the interspike intervals like? What are the firing rates? There's no way to tell the difference in our hands between the wild type and the null mice. So if you just think about these cells as indicators, if you will, of the animal's spatial representation individually, then the spatial coding as measured by these firing fields looks kind of like the behavior surprisingly normal in these animals, especially surprising given that they really do have pretty robust synaptic dysfunction. And whatever you think about it, this is not easily explained by this disruption hypothesis. But we had another hypothesis. That other hypothesis, which I've expanded on, that I call the discoordination hypothesis, asserts that you can actually get pretty reasonable behavior under simple circumstances by the individual actions, if you will, or the individual representational capacity of neurons. And the trouble happens when those neurons have to collectively gather their information in a coordinated way, in a reliably coordinated way, in order to do something more complicated. For example, switch between representing your location in the room and your location on the arena surface, or switch between recognizing that the shock zone is in the north and the shock zone in the south. And I will show you that is, in fact, the case. So let's look at timing in these FMR1 cells. Here's a image, initial stain from the hippocampus, and what I've overlaid on this is the local field potential that you can record at these locations relative here. And you'll see a number of facts. We don't have to dig into them, but they don't look the same. Although the spacing here, I think, is about 70 microns, right? So at every location within the hippocampus, the local field potential, which reflects in some sense the population synaptic activity, it looks pretty different. And why is that important? So to give you an intuition for the importance of that, let me tell you about my failures of surfing. So I don't know how to surf, and I've spent a lot of time thinking about surfing. So if you watch these three surfers, they catch the wave at the right phase or at the right time of the wave, and they use the energy of the wave to actually do something. All the other surfers are like me. We usually don't catch the wave at the right phase, and then the wave passes over us and we don't collect or use its energy in any way. So when neurons fire, they should fire at the right phases of these ongoing oscillatory signals. And I'm going to show you that that timing is actually abnormal in the FMR1 knockout mice. So this is an unusual plot, but let me walk you through it. On the x-axis represents the phase of an oscillation, and on the y-axis represents the frequency of that oscillation. So it's a collection of phase plots, and the z-axis, which shows the probability of the cell spiking at that particular phase of the particular oscillation. And so one way to think about it is if you summed across each row here, you would get the a probability of 1 for every row here. And you can see each cell, okay, here are place cells, here are some theta cells, here are cells that aren't place cells but are perfectly happy pyramidal cells. Each cell has its own characteristic, if you will, way of firing with relation to one of the ongoing or all of the ongoing oscillations. And that's characteristic of these cells. So we can make several measurements about that. If you look here at the spiking probability, that's what's in the color here. For the Fragile X mice compared to wild type, it's pretty normal. It looks just like the wild type in its relationship to the ongoing theta rhythm. The non-spatial place cells or pyramidal cells, these ones, are very unusual. They tend to have much lower likelihood of firing and having theta modulation compared to the wild type. The interneurons are, again, less modulated, and you see similar things with the gamma. So these cells have not as robust, in fact, a unusual set of relationships, weak relationships to these ongoing synaptic potentials, and we can dig into more of that. Here, I'll just skip this. If you were to calculate the pairwise discharge relationships amongst all the pairs of cells that you could record, and you organize them here, you can see that what's shown here are wild type and FMR1 knockout, that the organization tends to show, this is a little bit brighter compared to here, the organization shows that the knockout animals tend to co-fire together a little bit more than wild type. But what's even more remarkable is this is the first half of the recording, and the second half of the recording is very reliable in the wild type and not so reliable in the knockout mice. And that's a different sub-second time scales. It's not dependent on gamma or theta. So these cells, they discharge just fine, but they don't have reliable co-firing relationships. That's its baseline. What happens if you train them? After training in this task, if you go and you measure, let's look at the knockout animals, they tend to be hyper-correlated, if you will, much more experience-dependent tightening of their correlation patterns, so they tend to fire together more after this training, but their individual, remember, firing properties are pretty normal. So they, if you will, wire together persistently more. So what does that all mean? Okay. I'm going to describe and just wrap it up here that this causes what I'll call representational rigidity. When they build a place field type of map, if you will, of the environment, it's really hard to change it, and in fact it gets misapplied, and I'll show you that as we wrap up. I need to show you a cartoon for what I'll describe if you want to dig into the physiology of that. There are two reports here. If you think of the CA1 region, which we'll focus on, you can imagine that there are inputs from the CA3 region of the hippocampus, as well as inputs into the distal dendrites of the entorhinal cortex. These inputs in parallel recruit inhibition, and when the inhibition is recruited from CA3, we tend to have slow 30 hertz-ish gamma oscillations, and when the inputs are recruited from the entorhinal cortex, we have mid-frequency 70 hertz or so gamma oscillations. That's something we've described as well as others, and the significance of that is, let's look here. What's shown here in blue is a slow gamma oscillation on the descending phase of the ongoing theta wave. What's shown here in yellow is a mid-frequency gamma oscillation on the ascending phase of a theta wave. And these gamma oscillations preferentially have these phase relationships. And if you look here, what we can describe is something called slow gamma dominance. I won't dig into that, but all that means is that in a particular theta cycle, there's going to be more power in the slow gamma band than the mid-frequency gamma band, and that will be what we'll consider dominant. And that turns out to be rare, but you see it. Okay? The consequence of that is shown in this 5 seconds or so example. Let's imagine you've recorded 6 place cells. Okay? Slow gamma dominance, the magnitude of it is shown here in red. It's simply the ratio of the slow power over the mid-frequency power. And if you look here, the animal is over here. What's shown here is the Bayesian posterior, the likelihood of where the animal is given the population activity. I think in this case at 300 millisecond resolution. And so the animal's in this location. And what we observe is when the animal's in that location, these cells that have corresponding firing fields tend to fire. As the slow gamma dominance goes up, these cells tend to not fire. Notice the animal is still in the same location. And now these cells, 5 and 6, tend to fire. Their firing fields are close to the shock zone. And in fact, that is where the posterior points to. The animal is here, okay, but its place cells are signaling some other place. And then a few seconds later, the posterior catches up and you get local decoding again. Why that turns out to be important is because as slow gamma dominance is observed, the firing becomes non-local, right? When slow gamma dominance is low, firing is local. When it is high, as in this case, discharge tends to be non-local. And so you can see, or I'll show you now, the consequence of that. Here's a wild-type animal, and at time 0, the animal is going to turn and walk away from the shock zone and make its avoidance. And we're going to look at, 6 seconds before and 6 seconds after. So 6 seconds before it turns and walks away, the decoding is local to where the animal is. About 2 seconds before the animal is going to make its avoidance, then we have decoding and activity now pointing to the shock zone and where the animal is going to go to. Okay? At the moment the animal turns around, again the decoding is split here. After he's turned and walked away, the decoding catches up and becomes local again. So these animals are using, or the slow gamma dominance is causing the network activity to change where the representation, if you will, points to. Here's another example. And when the animal makes an error and enters the shock zone, you'll see in this wild-type case, the animal doesn't make the avoidance, and the decoding stays local to where the animal is. He's run away, and it actually looks back at the shock zone. Okay? If you simply ignore whether the animal was avoiding or not and calculate where was the posterior when there was slow gamma dominance, you'll see that when there's slow gamma dominance, the posterior points to the region of space that the animal's actually avoiding, and not so if you just pick random periods of time. If you look at the knockout animals, what you can see is, I remind you that when we put the animal in the conflict case, its performance was impaired. And we see that slow gamma dominance in the knockout animals actually is very elevated in these cases when the animals are unable to do the conflict, whereas the wild-type animals slow down the slow gamma dominance, which one of our papers demonstrates is actually the animal recollecting the shock zone. And you can see that here, and I'll wrap it up. Okay? Here's the wild-type animal. I'll ignore that. The knockout animal is very similar in the case where the animal is avoiding the initial shock zone. What's interesting is when we've changed the shock zone to the conflicting location over here. The wild-type animal, when it enters the location of the former shock zone, it is going to run away at this moment. The activity just preceding that points to the current location of shock, and then the animal will leave. Okay? If you look at the knockout mouse, this is typical. The activity is local. 2 seconds before it's going to run away, the activity is still local. In fact, is pointing to the current, to the former location of shock. And then as the animal walks away, it becomes local to where he is and is going to go. And in the summary, you can see here that in the wild-type animals, the probability of decoding where the animal is pointing to the conflicting, to the current shock zone on the conflict test, whereas the knockout animals, when they mean to avoid, they think about the former initial location of shock, or at least they decode to that reliably, which explains why they perseverate and they can't actually learn the new shock zone as quickly. So what have I told you or showed you about these mice? They have experience-dependent synaptic plasticity abnormalities. Their temporally discoordinated spike discharge seems very prominent, and you can demonstrate it in many ways. I just talked about their representational rigidity, at least within the hippocampal formation. And in many ways, these are the consequences of loss of FMRP. However, there are really very few changes that you can demonstrate in the cell individual properties. I also showed you that if you wanted to change the network function, you could use this cognitive control training, right? Train the animals so that they have to ignore something irrelevant, and that makes very long-lasting changes in this circuit. So one of the things that we are working on is, could we use this kind of training in a circuit-specific way so that if we were to appropriately curate the cognitive training to adjust these circuits, could we persistently change the information processing in these knockout animals in a way that is amenable to the particular changes that we've seen, such that they can actually learn, if you will, to learn better, and learn in particular to meet the challenges of having information contradict what they currently do. And so that is the current state of affairs with this research program. And we mean to use various agents and stimulation opportunities to make the system more and less plastic at the appropriate times of the curation. So with that, I thank you for your attention. And the people highlighted in red contributed to this, as well as the folks who funded the work that I presented in red. Thanks for your attention.
All right, time for some questions. Questions? This question back to you.
Yeah, well, I guess I have to ask this. Have you investigated these knockout mice with neurons response during replay, or did you sleep? Do they have like similar?
We have not looked at them during sleep at all. And the main reason in fact, we even have recordings of them during sleep. The main reason we haven't is those recordings were really designed for us to study the response during these events that cause the slow gamma dominance. And the trigger for that slow gamma dominance, I didn't talk about it, is an event called a dentate spike. And so those dentate spikes appear sometimes during sleep, but very often while the animal's walking around, and that happens to be the thing that we're focusing on, rather than the replay, because the dentate spike, the synaptic plasticity change that I described affects the consequence of that dentate spike. And the dentate spike is setting the slow gamma dominance through the entire trisynaptic pathway. So there's more to it than simply looking at replay, because the circuits actually change.
Quick question. So towards the end of your part one, you showed some measurements of in vivo synaptic activity, these sigmoidal curves. So you showed what happens to inhibition, but did you also measure excitation?
Yes. The rest of that paper does. So what's very, very interesting is your impression of the synaptic change from a hippocampal slice will be absolutely misleading, and it's the opposite of that. So if you look at a hippocampal slice, where I want to point out in a hippocampal slice, there's no ambient ongoing activity to drive inhibition, first of all. We don't block inhibition when we do these experiments. You will see a synaptic potentiation. Okay? And we can see that potentiation for at least a month, okay, after training. We've published that a number of times. If, however, you leave the brain intact and you make the same measurements, what you see globally is a depression of that synapse. Okay? And we used optogenetic stimulation of inhibitory neurons to mimic those changes, which we could do. And what we discovered is if you look at the very short time scale responses, is I showed you the cartoon of the actual circuit, there's something complicated that happens. There's a global inhibition, explaining what you saw, but there's also a pathway-specific local disinhibition that improves the synaptic strength on that pathway. So if you block inhibition or there's no ambient activity to drive inhibition, all you see is the potentiation and you miss all of the very interesting inhibitory plasticity. Okay, you can see that that paper is all about what I just said. Okay, the Chung et al. Paper. Hey.
A lot of information, Andre, but I have a point of clarification, perhaps. So you showed that the place fields of individual neurons in the FMR1 knockout mice do not change from wild type and may even be better.
Yep.
Yet the phase relationship of single spikes firing in relation to the ongoing rhythm, perhaps theta. So my point is this, that isn't the phase relationship between individual spikes and the underlying theta a critical driver of place field formation via spike timing mechanisms? You show a disruption in the phase relationship, yet no disruption of the place field. Did I get that right?
Absolutely right.
And what is your interpretation of this?
I'm going with the data. So it was a belief that those phase relationships were important because we see them and they persist. But these data show that those phase relationships can be, they're not completely gone, but they can be severely weakened. And you still get, in fact, you get better actual place field tuning, slightly better. Like I could statistically demonstrate it, but it's marginal. Okay? So I'll give you my interpretation of that. But first I want to show you, look at this plot. The cells that you would classify as a place cell that have a nice firing field that you would look at or that we could quantify and say that cell has location-specific firing, they have a normal firing relationship with theta. See that? And with gamma. Okay? But those are only 20, maximum 25% of the population. The rest of the cells, they are not to be ignored by the brain, presumably. Right? We ignore them, but maybe we shouldn't. They have, these are the non-spatial pyramidal cells. They have this abnormal relationship. So do the interneurons. Okay, so one interpretation is that we haven't been wrong in a sense. Place cells actually, in order to have their location-specific firing, are abiding, if you will, by those phase relationships, right? Normal here, at least with theta. Okay. But it's the other cells in the network that aren't well coordinated and that might be helping to explain some of the activity, some of the dysfunction that we see. To me, that's a very interesting opportunity because it suggests that there are populations of cells, quite a lot of them, that are open to manipulation to see if we can get them, if you will, to fall into the right timing relationships. Maybe not by instructing each and every one of them, but by relaxing and then allowing the synaptic plasticity to operate under conditions where we help to control that, under behavioral conditions that we provide, if you will, lovingly to the mice to give them an opportunity to learn this world that they're pretty capable of if you present the information to them properly, right, in a way that's generous. Does that make sense? That's the thing we're exploring.
Last questions?
I have a question. Actually, I have two questions. So
You're in charge.
So when I kind of think about this, I think back to some of the early work, like the Tonegawa work looking at NMDA receptor disruption, in particular in the shape cloud. So this was like in CA3, disrupt synaptic plasticity of the shape cloud projections. You get a very similar phenotype, and that is that the CA1 place cells seem normal. They had normal reference memory. And a lot of the tasks that you kind of described, remembering that part of the room is bad. Sort of what we call reference memory task, right? It's bad now, it's bad tomorrow, it's bad.
It's always bad.
And in these CA3 NR1 knockout mice, normal CA1 place fields, normal reference memory, there was a deficit in working memory, what we describe as working memory. That is sort of a trend within a room, you sort of change the rules. So now they're looking for a hidden platform, you move the platform. So they have to remember, I used to be over there. So they have a working memory deficit. And that's where you also saw a place field deficit. So there was a transient disruption in place field and working memory condition were disrupted. Interestingly, kind of relates to Jack's question, that and after the working memory perturbation, the next day after a period of unmonitored sleep, the place fields seemed normal. So it seemed to be like there was something going on during sleep that sort of covered them. Anyway, these phenotypes seem to be associated with a CA3 specific plasticity disruption, very similar to this. And then I'm thinking about this is the sort of the gamma, the slow
Gamma dominant.
Slow gamma dominant. So the slow gamma dominant is the CA3 component. And the CA3 component, I sort of describe this, you do the decoding, I sort of describe these hippocampal theta sequences. Within theta, the place representation is not stationary, but it actually kind of sweeps from present location to future location, present to future. That's also associated with this phase-shifted, the entorhinal CA3 component. So like CA1 place cells are being driven by first entorhinal, then CA3, entorhinal CA3. And the CA3 component is the one that's sort of prospective by a couple of seconds. It maps out to where the animal's going to be like 2 seconds later. And that's sort of similar to what you're actually showing here, that shifting the dominance between like entorhinal and CA3 would affect this theta sequencing. So that my super long-winded question is, theta sequencing disrupted? As you see a disruption in this process, and that would also kind of relate to the question of phase disruption. Is it really just general phase disruption, or is it disruption of the CA3-driven prospective component?
So theta sequencing appears disrupted here. Okay? And it's however complicated, we've never published it, because I think the right way to do that is to have the animals run on a linear track so that you can be confident about what phase the next spike should appear on, which is difficult in these open field experiments. But my investigation of that suggests that it's the right. It seems stuck, right? So, and very often it's stuck in a like here and now way. A little bit ahead, but like that.
So
but it's very difficult to say because the animal's not running, reliably in a straight way.
So you think that really is like a CA3 plastic, something about it, disruption of CA3. I mean,
Yeah, it seems to me.
You could do through NR1,
Yeah, I think there are many ways. If you built a system that was plastic and you built it so that you couldn't regulate that plasticity easily, and you made it such that it was hyperplastic, then it would get stuck in the first way it got tuned. These animals behave consistent with that interpretation. And the neurons sort of behave, they more or less behave consistent with that. And so in order to test that, what you have to do is manipulate plasticity, right? So you can make them less plastic, if you will. We happen to study a molecule called PKM zeta that is a mechanism for that enduring plasticity. And so instead of being disruptive to these animals, one would make the prediction that if you were able to destroy PKM zeta, in the Schaefer collateral region, in a subset of the places, you would actually turn these FMR1 null mice into normal mice, which is an explicit prediction, and we're trying to do that experiment.
Or thinking of it as like, it's a CA3, it might be a CA3 specific. Okay. But
I think
dominated
there's
by
a,
CA3
it's
plasticity.
dominated by CA3
Plasticity.
plasticity and activity drive. And that's not to say one doesn't happen, but it's dominant. That's our working guess at this point.
And the last question, too long-winded, so I think we're,
There is a question on YouTube.
oh, YouTube, right.
By somebody called PZ Lemons.
Right.
Okay?
PZ Lemons?
Well,
My pleasure.
that's their handle. Okay. In FXS model mice, is there an attenuation of the absolute magnitudes of non-spatial and interneuronal hippocampal activity? Or is it normal?
It, to the extent that we can measure it from individual cells, it's normal. We've not been able to measure a difference. And we've,
I hope your questioner heard that. I think they did. All right.
Okay, we're
Okay.
done. I guess there, we have a reception, is that true?
Yes, of
Yes,
course.
it is true, the reception.
Always.
Please, everybody join us.
That is, yeah.
thank you.
Thinking about phase perception, the sequencing emerging as a result of behavior and requiring actually correlative activity, some kind of
Well,
plasticity preference.
so
You get phase perception.
you can kind of get it.